Hello friends, this is Dr. Rajiv Ranjan and I am here to tell you about PCR, that is called polymerase chain reaction. So polymerase chain reaction is the very important technique, or somebody can call technology, to be used to amplify a single piece of DNA by a series of heating and cooling cycles, generating millions of copies of particular DNA sequence, particularly in vitro. Means you can generate billions of copies in laboratory conditions if you have template DNA, if you have primers, if you have DNA polymerase, and if you have nucleotides. So these are the components of PCR. I told you DNA template, the DNA region, the target to be amplified, DNA primers, that is also known as oligonucleotides, that is around 20 to 24 nucleotides, and it will help to amplify the target DNA. And third is called DNA polymerase, that is called heat stabilized polymerase and synthesize the new DNA. And fourth is nucleotides, that is the building block of DNA, like we are calling DNTPs. We have DATP, then we have CTP, then we have GTP. Likewise, you have all the 4 DNTPs, and these are the, and apart from that, you should have some buffer, then you should have distilled water, and you should have PCR machine. So you see, so these are the PCR machine, that is called thermocycler, and if you set up the reaction in small PCR tubes, and if you keep the PCR tubes in this machine, finally you will get billions of copies of particular target DNA. PCR machine may be different types. Company are providing, like you have BioRed, you have Agilent, you have thermocycle, that is called Agilent, Appendoff. Okay, so now if you see the history of PCR, so PCR was invented in 1984 as a way to make numerous copies of DNA fragments in the laboratory. So already I told you this is nothing but it is an in vitro DNA replication. So whatever you have seen in DNA replication in vivo conditions, this is the in vitro conditions. We are generating billions of copies of target DNA in laboratory conditions, just putting the or setting up the PCR reaction and putting into the machine. So beginning with single molecule of genetic material of DNA, the PCR can generate 100 billion similar molecules in just few hours, maybe 2 hours, maybe 3 hours, depending upon the ramping rate. Now, this is the person that is called Carey Mullis, and he is the person behind the PCR, and for that he got the Nobel Prize in 1993. That is called Carey Mullis. So now we will see what is the basic principle of PCR. So I told you PCR have 3 basic steps. We have denaturation, then annealing, then extension. So denaturation means we are heating the PCR, that is called template, at 94 degrees centigrade. Annealing, where RNA polymerase, where RNA or DNA primers or oligonucleotides binds. Extension, here DNA polymerase will come and extend the primers. So we will see in detail. So if you see the these are the reactions. So we have DNA template, that is called double-stranded DNA template. Then if you heat it, it will get denatured. It will be separated into 2 single-stranded DNA. Then if you anneal it, primers will bind to the single-stranded DNA, and if you extend it at 72 degrees centigrade, then the chain is going to be elongated. So now in that you can see, if you see the movement condition, you can see how the double DNA strands separating into the single-stranded, and how oligonucleotides, they are searching the complementary strand and bind to the where they will find the complementary, and finally you see DNA polymerase is coming and extending the further reactions, that is called extension. So we have all the 3 steps in video form. We have denaturation, we have annealing, and you have extension. So now you can see how beautiful this video is running. So now come to the next slide. So after amplification, you have to verify the PCR product, whether PCR product has been done properly or not, or performed properly or not. So you can see the diagram. These are the gel pictures. So these are the in first band. This is the DNA ladder, and these are the all PCR bands. So we are measuring. We are just measuring the size, how much length of PCR. This is the PCR fragment length by what we call matching with the DNA ladders. So now we see what is the application of PCR. So I told you PCR is one of the basic tools in molecular biology. So if you see, PCR can be applied into various techniques like electrophoresis, southern blotting, enzyme linked hybridization, cross-linked hybridization, FISH, that is called fluorescent in situ hybridization, then allele specific oligonucleotide hybridization, then direct sequencing. So these are the different applications of PCR in various techniques. Now if you see application of PCR as per the field of science, so PCR can be used in medical application, in infectious disease application, in forensic science application, like DNA fingerprinting, one of the major what call nowadays every what call in forensic sciences persons or experts are going for the PCR. Then research application, and research application nowadays for cloning, nowadays for molecular marker, for various processes. If you want to apply certain DNA fragments, if you want to amplify certain genes, if you want to amplify certain plasmid DNA fragment or chromosomal DNA. So in research there are n number of uses of PCR. So these are all about application of PCR. Thank you very much.